Endoplasmic reticulum, the next organelle, also known as endoskeleton of the cell. ER name was given by Porter in 1961. It is present in all eukaryotic cells and it consists of A. Cisternae, long, flattened and unbranched units arranged in stacks 40 to 50 nanometers thick. Vesicles, 25 to 500 nanometers in diameter. Tubules, long, isolated, branched units, 50 to 190 nanometers thick. The structure of endoplasmic reticulum is like the Golgi body, but in endoplasmic reticulum, cisternae, vesicles and tubules are isolated in cytoplasm and these do not form a complex. Now you can see here the rough endoplasmic reticulum and the smooth endoplasmic reticulum, and you can see they are associated with the nucleus. Golgi body is a localized cell organelle, while endoplasmic reticulum is widespread in cytoplasm. Types. ER consists of two types. Endoplasmic reticulum consists of two types. Number one, rough endoplasmic reticulum or granular endoplasmic reticulum. Granular means that endoplasmic reticulum associated with the ribosomes. A smooth endoplasmic reticulum or agranular endoplasmic reticulum. Now you can see these ribosomes attached with the rough and no ribosome in the case of a smooth endoplasmic reticulum. Now let us take the difference. Rough ER, that is rough endoplasmic reticulum and a smooth endoplasmic reticulum. 80S ribosome bind by their larger subunit with the help of two glycoproteins which occur on the surface of rough endoplasmic reticulum, that is riboporine 1 and 2, and they are more stable. Ribosomes and riboporines are absent in a smooth endoplasmic reticulum. Rough endoplasmic reticulum is more stable, while smooth is less stable, composed of cisternae and vesicles, that is rough endoplasmic reticulum, abundantly occurs in the cells which are actively engaged in protein synthesis, say for example liver, pancreas, goblet cells. While smooth endoplasmic reticulum composed of tubules abundantly occur in cells concerned with the glycogen and lipid metabolism, say adipose tissue, interstitial cells, muscles, glycogen, storing liver cells and adrenal cortex. Maximum number of rough endoplasmic reticulum present in the cells of fibroblast, formation of fibers or protein synthesis, plasma cells associated with the antibodies formation, pancreatic cells associated with the enzymes which consist of proteins. Maximum number of smooth endoplasmic reticulum present in the cells of interstitial cells, that is Leydig cells, found in testes. Functions is to secrete testosterone. Cells of the adrenal cortex, synthesis of steroid hormones. In muscle fibers, and in muscle cells, these smooth endoplasmic reticulum is referred as sarcoplasmic reticulum. It is rich in calcium ions and energy-rich compounds required for the muscle contraction, and they passes impulses from the surface membrane to the deep region of the muscle fibers. Here is the sarcoplasmic reticulum. T-tubules. These transmit stimulus of contraction inside the muscle cells. Ergastoplasm. Modification of rough endoplasmic reticulum. Ergastoplasm of nerve cells called Nasal Bodies. They do not exist as such in the living cells. They are used in vitro study of protein synthesis. Glucose 6-phosphatase is one of the marker enzymes of endoplasmic reticulum. The other enzymes which are associated with the synthesis of triglycerides, phospholipids, and cholesterol, NADH diaporase, NADH cytochrome C reductase, magnesium ion activated ATPase, nucleotide diphosphatase, ascorbic acid synthetase, etc. Functions of ER. Mechanical support. This is one of the main functions of the endoplasmic reticulum. It participates in the formation of cytoskeleton with microfilaments, microtubules, and intermediate filaments. Intracellular exchange helps in the transport of materials in cytoplasm from one place to another, and it occurs through the ducts of endoplasmic reticulum. Rough endoplasmic reticulum participates in the protein synthesis. Lipid secretion. Lipid synthesized by the agranular portion of ER, that is a smooth endoplasmic reticulum, is stored into the Golgi bodies before finally being extruded into the cytoplasm as lipid droplet. ER elements are arranged around the chromosome, and they help in finding or in providing a new nuclear membrane after every division. You can see here helping in the formation of a nuclear membrane. Formation of cell plate, formation of lysosomes and Golgi body. And here you can see the smooth endoplasmic reticulum is concerned with the detoxification of drugs and steroids. Plastid. The term plastid first used by Haeckel in 1865. On the basis of the function of plastids, we categorize them into three types, that is leucoplast, chloroplast, and chromoplast. Leucoplast. And they, on the basis of their function, because they are concerned with the food storage, again divided into three types. Number one, amyloplast, leucoplast, which is storing starch. Erythroplast, the leucoplast is storing proteins. And you can see amyloplast with starch and erythroplast with proteins. Eryoplast, leucoplast is storing oil droplets and fats. You can see. Chromoplast means other than green are referred as chromoplast. These are colored plastid responsible for color of flowers, fruits, and algae. In algae, brown color is due to puoplast and red color is due to rhodoplast. Chromoplast occur mainly in the pericarp and petals. In flowers, you know that chromoplast is helping in pollination, and in fruits, they help in the dispersal of the seeds.